In this video, we will discuss about adeno-associated virus transduction. We might have covered this video in our previous videos. However, we will cover these videos again and we'll go through some of the aspects that we have not covered in our previous videos. These are the adeno-associated virus. So it internalizes in the cell with the help of these glycosylated cell surface receptors. That is, adeno-associated virus interacted with these glycosylated cell surface receptors that helps in the endocytosis or internalizations of this adeno-associated virus, and thereafter it will go through this early endosomes with the help of cytoskeletons, and with the decrease in the pH as it goes from early endosomes to late endosomes, the adeno-associated virus escapes from this capsid, and thereafter it can either go inside the nucleus with the help of through this nuclear pore entry, or it can also goes to this proteasome where they will be degraded into the peptides. So for the proteins that will, or for the adeno-associated virus that will go in the nucleus through this nuclear pore entry, will then go into will then go towards, or will then proceed towards these uncoating steps. Thereby it can either go towards this thing, thereby it can either go in this way or in this way. This depends on the kind of genetic informations this adeno-associated virus contain. If it contains the single-stranded adeno-associated virus, it can go in this way, and since it is a single-stranded adeno-associated virus, then at this point of time it cannot be used for transcription, since it is not transcription ready. For transcription, it needs to be double-stranded DNA, and therefore single-stranded DNA will go towards this double-stranded DNA formation or second-strand synthesis, and thereafter it can go for the transcription and then translation. And similar thing can happens for this 3' this negative strands. So the single strands can be either positive strands, that is 3' to 5', or it can be negative strands, that is 5' to 3'. So in this case, the complementary strand synthesis needs to be occurs, and these things can be occurs through the strands annealing and the second strand synthesis formations, and thereafter it can go for transcription and translation. And in other case, if there is self-complementary adeno-associated virus are present in the this adeno-associated virus, if the adeno-associated virus contains this self-complementary DNA, then it will has the both negative strands and the positive strands, and in that case it is ready to go for transcription and translation. And while going for transcription translation, it can go in different forms. First, it can form a single circular monomer where only a single strand of the DNA is attached, or it can form a circular dimer where the genetic information is attached at two part of this circular DNA, or it can be the circular contamer where the genetic informations or gene of interest is attached at multiple sites. So in most of the cases, when the genetic information is introduced to the adeno-associated virus, it can form one of these structures, and there are very less possibilities or very less cases where this genetic informations or this DNA is integrated into the host genome. Therefore, it is shown here as a dashed line. So what is the advantage of this adeno-associated virus? Its advantage is that we can transduce a gene of interest within the cells without disturbing the host genome to a greater extent. Whereas when we use the lentivirus, the possibility of this integration into the host genome is very high, because the lentivirus works in a way that the gene of interest is integrated into the host genome. And That is the reason adeno-associated virus is used for vaccines compared to the lentivirus, because uh, people don't want to disturb their genome that might have some adverse effect later on. So I think it's beneficial to have additional circular DNA structures that can be used for the replication of this gene of interest that has been transduced through this adeno-associated virus. So I hope you got a basic idea about the adeno-associated virus transductions and why it is beneficial compared to the lentivirus. And this beneficial part I might not have covered in our previous videos. That's why I have again explaining this adeno-associated virus transduction process, so that people can understand why this AAV is preferred over lentivirus for vaccines manufacturing. That's all for this video. Thank you for your time.